So this morning we talked about intro to sequencing, processing the data, calling variants from a high level, both germline and somatic. No, germline, and now what we're going to do is the somatic variant discovery overview. So I'm going to give you a little run through of the main challenges that are involved in somatic variant discovery, and I will say this is mostly focused on cancer. There are forms of somatic variation that are obviously not cancer. Cancer is kind of the extreme form when things really go wrong. But we're going to focus on cancer because that's kind of the most common use case that we deal with. And so I'll talk about what's more challenging about somatic variant discovery compared to germline, and what are the adaptations that we make to the workflows to compensate. After this, we'll talk about actually how do we assemble all of these things into pipelines, how do we run pipelines at scale, and all of that good stuff. But for now, let's talk about somatic variant discovery. All right. So this is a kind of a schematic depiction of what happens over time within the context of a tumor. So if you, and I should say also the little, so this is over time. So as we go further towards the right, the tumor is aging and things are happening within it. And the little lightning symbols are mutation events. And so if you think about what's happening from that single key mutation event that kicks off kind of the chain reaction that leads to tumor development, there's a variety of things that happen within this tissue, where you start with one cell that mutates, and you end up with a kind of cascade of events that leads to a tumor of many cells. And within that tumor, you have multiple lineages of cells that start to arise as you have individual mutations that happen, that occur. Some of them will be just kind of neutral mutations that just happen, but they don't really have any effect, don't really have any consequences. But then some of the mutations that happen, the ones that are depicted in red, those are going to be driver mutations, and they're called driver mutations because they drive the evolution of the tumor. You'll have some of these mutations have an effect on the cells that they're in, that give them a sort of evolutionary advantage. You can think of it as a sort of evolution in a petri dish, because you'll have one lineage of cell that suddenly has a particular advantage in terms of how quickly it can proliferate and how it can potentially resist efforts to combat it, resist therapeutic effort. And so you'll have the kind of appearance of these sub-lineages of cells within the tumor that will progress to various, in various proportions. And as you have other driver mutations that occur, you have new progeny cell lines that appear within that context. And so as you go, and as that gets more complicated, you can imagine that the tumor itself becomes more complicated, and when we're taking a sample of tissue to sequence it, we're not sampling one population of cells, we're sampling multiple populations that have different mixes of mutation. And that is where most of our headaches are going to come from. Because it means that let's say we sample at this point, we have these multiple populations, and so when we're ultimately looking for a variant, often that variant is only going to be, especially if we're looking for driver mutations, they're only going to be present in a subset of the cells that we're sampling. And so we're no longer going to be able to rely on the classic expectation of oh, I'm a diploid organism, so either all of the reads support my variants or half of them support the variants, right? Now that expectation goes out the window, and I will have different allele frequencies, different levels of support for the variant in the data that I'm generating from the tissue. So a lot of the complexity that we deal with in somatic contexts come from there. Does that make sense? Okay. All right. So we're going to be looking for these driver events, driver mutations. If we take a step back and think about what is involved in the appearance and proliferation of tumors, cancer, there's a number of different things that cause cancer, right? Coffee, bacon, all the good stuff. Also some of the bad stuff. In terms of lifestyle factors, you can think about effective radiation, carcinogens, chemicals that we put inside our body voluntarily or involuntarily that we might be exposed to in our work environment or in the living environment. So there's a number of things that are just coming at us. There are some cancer determinants that also may come from our genetic code. There are some things that we carry that may predispose us to developing some kind of cancer. There's a lot to look at, but the big differentiation I want to make here is that some of these things are part of our normal germline variation. These are not the things we're looking for here. What we're going to be looking for here are the driver mutations that are happening kind of in real time as we age as an organism. So we're going to want to exclude the normal variation. And we want to look at what are the somatic alterations that are happening in the tumor in response to all of those factors, but the things that are happening to us that are not things we already carry. Does that make sense? There's a distinction? Okay. And so the key model that we're going to use for identifying those variants, that we're going to do variant calling on a sample that's taken from a tumor, and we're going to compare that to the variant calls we get from a normal. And that's your classic tumor-normal pair. And that pairing is designed to allow us to differentiate what is newly acquired in the genome, these driver mutation, distinct from the original background variation that we already carried. Okay, any questions so far? Nope, straightforward. All right. Okay, so, yes, sorry about that. Okay, so we have set that context. Now there's a complicating factor, right? Because I just told you it's super simple. You just sequence normal tissue that's unaffected by the cancer, sequence a tumor, kind of do a subtraction, and whatever is unique to the tumor is your somatic mutation load. The problem is that when you sample tissue, it's rarely so clean cut. It can be very difficult to have pure normal and pure tumor, because as your tumor develops within the tissue context, there's a lot of biology that's happening. Part of it is that as your tumor evolves and you have those various cell lines, again, there's that heterogeneity that I mentioned earlier. That means that some of the new mutations are present in some of the cells and not others. At the same time, you have some normal tissue that's mixed up in your tumor. Depending on how you're taking the sampling, and honestly the skill of the pathologist who's making the sampling, you can have more or less normal tissue that gets that contaminates your tumor. And at the same time, you can have the same problem, the reverse problem, I should say. Sometimes cancer cells migrate and end up in in your normal sample. Most analyses are done with matched blood normals. So you take blood sample to take the normal tissue. Unfortunately, sometimes you have circulating cancer cells that cause some contamination of your normal. And of course, you have the problem of blood cancers, blood-borne cancers, where like the blood itself is your cancer tissue. And so we have to deal with all of these kind of case figures. I'm mostly going to focus this on what we do for when you have a solid tumor sampling and how we deal with contrasting that to the normal. And I will just say that for most blood-borne cancers, what we end up doing is tumor only. We often don't have a good normal to use for those kinds of cancers. So in those cases, we tend to resort more to tumor-only analysis. And I'll have we'll talk briefly about that. But most of what we're talking about here is tumor normal, all right? All right, so you've seen that slide already. You're probably getting sick of it. I will remind you that we're looking at both the short variants, SNVs and indels, but also at the copy number variation. So I'm going to show you two different workflows. One workflow is for the short variants. One workflow is for copy number. In both cases, we have to deal with this problem that if we sum up all the sources of noise, the biological and technical sources of noise in the data that we have this problem if we compare somatic to germline analysis, that in germline we have that nice expectation that I mentioned of a nice strong signal where you have ploidy expectation and the amount of technical noise is usually pretty small. When I say noise, I mean the artifacts and errors that creep in. So we usually have a pretty good differentiation for most of the data. Most variant calls are pretty easy to make. But in the somatic context, between the heterogeneity problem that I mentioned, other types of mutations like the compounding effect of copy number on top of short variants, which is much more common. And also the issue of contamination, of having contamination within the sample, but also the effects of cross-sample contamination, which I'll talk about briefly. You just have a lot more noise and a lot more error. And so you need a pipeline and a variant calling model that is specifically tailored to that. And that is why you cannot use a germline variant caller to call somatic mutations and vice versa. So we don't recommend using a somatic caller to identify germline variation, just because the modeling is very different. All right? Okay. All right. So just to recap the kind of problems that affect us in the somatic context, you have all the usual, which the grayed out ones are the things that affect germline and somatic equally. So we've talked this morning about batch effects. We talked about some of the artifacts that come up during sample preparation. Andre talked about the oxidation that can happen during the shearing of the DNA. But there's also a few other problems that we need to take into account, like sample swaps. This is something that happens in all cases. If you're swapping your samples, like literally mislabeling samples, you want to think it doesn't happen, but it does. We've seen some projects in the past where we sequenced, we processed samples for an external collaborator who will not be named, where there were a lot of sample swaps, just samples that had the wrong identifier. In some cases, we were able, after the fact, to re-identify the samples because we had complementary information about those samples. In some cases, we were not able to, and those samples just had to be thrown out. It was just a waste of time and money. So that's something we have to pay attention to. But now in the somatic use case, on top of that, we're going to have these additional sources of problems. One is cross-sample contamination. One is the sequencing artifact. The impact of sequencing artifact is going to be much greater. These are things that can affect germline sequencing too, but they have just less impact in a germline context because cross-sample contamination, I'm talking about a little bit of DNA from one person ends up in the tube from another person. Again, you want to think it doesn't happen, it happens. And most of the time, this is at a very small level, a small enough percentage of material that it doesn't matter for germline calling because the reads from the other person, there's only 2 or 3 at a given site, and so we just dismiss that as being error, as being background noise. But in the somatic context, that might be potentially an allele frequency where you need to be able to retrieve actual somatic variants. So that's going to be more of a problem. And same with sequencing artifacts that appear only at very small frequencies, so you don't have to worry about them in germline context. For somatic, you actually do have to worry about them because that might be the level at which you need to retrieve somatic calls. And then on top of that, the things that I talked about in the first few slides of the talk, with the problem of not having enough purity, so having tumor normal contamination, the heterogeneity of the tumor since it evolves over time, and then the compound effect of having several mutation types affecting the same regions of DNA. So all of that means that it's a lot harder, and that's why you have dedicated tools and more kind of robust approaches to dealing with contamination. And this is kind of just a very detailed view of the different types of information that will come into the analysis of somatic variation, where you have multiple things that you have to take into account. They're typically more complex than in the germline context. But I'm not going to go into the detail of this, but I just want to remind you that in a cancer context, you will have some alterations, some somatic alterations. They're much more dramatic than what you see in the germline context, because if you see like a karyotype for a normal cell, that's the karyotype that you kind of expect, right? But then you look at a cancer cell line, and admittedly cell lines can be a little messed up in other ways, but you can see that the alterations that affect these chromosomes in the cell line are really dramatic. You can have multiple copies of some are entirely missing. You have some rearrangements where pieces of different chromosomes are grafted together. That's going to have a big effect on what the data looks like on the other end. And this is obviously something that you don't see in the germline context because that would not be viable. Like you would not have a viable organism with this genome. But in this context of a tumor, it's weirdly extra viable. It's making that tumor extra able to proliferate because of the effect of some of these alterations. Okay. All right, so that's kind of the context and the challenges we need to deal with. So let's look at 2 workflows. The one on the left is a short variant workflow. It's fairly complex, and we'll go into the detail tomorrow. I'll go just through the top level steps in the next few slides. Then after that, I'll give you an overview of the somatic copy number variants discovery workflow. All right, so let's focus on that one first. This workflow relies entirely on, well, almost entirely, on the tumor normal comparison. Like this is the strength, the best way to run this is if you have a tumor and it's matched normal, so tissue from the same person, from a healthy part of their body, you can make this very simple but very powerful comparison of looking at any given site, like at this site. In my tumor, I see a few reads that support a possible mutation. But then I look at what's in my normal, and I have reads supporting that variant as well. And that tells me, well, that's very likely to be just a germline variant that is part of my normal background. So I can rule that out as being a tumor, a somatic mutation. However, here at this other site, I have a few reads that have some mismatches. I have nothing equivalent in the normal, so it's reasonable to say maybe that's a somatic variant. Of course, that also could be just sequencing error or library prep artifact, right? But at least this gives me a first point of anchoring for eliminating some of the most obvious stuff that's not somatic variant. If you don't have a matched normal, you can still run what follows. The problem is that you will not have as much power to eliminate the normal variation, and you'll just have to do extra filtering. But having that normal available is very important. And there's the slide that explains it. Now, if you're going to do that, you need to have, in all cases, you want to have something called a panel of normals. It has to be extra good if you're doing tumor only. Because the panel of normals is going to help you eliminate that stuff. Now let's talk a little bit about panel of normals. The panel of normals is a reference panel that is ultimately a list of variants that you generate from a bunch of normal samples. They can be completely unrelated to the subjects in your analysis. And the idea is to take samples that were sequenced, generated with the exact same technology, processed in the same way, and you actually generate the panel of normals with the same somatic variant caller. And it's basically saying, what would the program give me as somatic variation if I ran it in tumor-only mode on a bunch of normals? And the goal is to understand what is the background noise that is reoccurring across many normals, because we know that a lot of these artifacts can reoccur in the same place in many samples. And so with the panel of normals, we're trying to capture that background noise. It's important to understand you're not actually trying to capture normal germline variation with the panel of normals, although you can also get that as a kind of a resource of known variants. But the primary goal is really to eliminate technical artifacts that are like systematic errors. But for that, it's important to use as normals. You have to process them in the same way as your cancer sample. It has a borderline effect of also eliminating germline variants, but again, that's not its primary purpose. Does that make sense?
Say more about what is called unrelated normal. Usually we do that in normal tumor care.
Mm-hmm.
Can we use that?
So you can use, like if you have a bunch of matched normals for your tumor samples, you can use those matched normals as part of your panel of normals. But we do recommend using more, like additional other matched normals if available, because ideally you want to have as many of those as possible. As many normals that are technically applicable, because they're generated with the same technology, as many of those as possible included in your panel of normals.
So it's better use the normal, not related to my...
Well, you can use the related ones, but usually that will still not be enough samples to make a robust panel, and at that point then you just take, if you happen to work in a facility that also does germline sequencing, that's what we do. So we have access to a whole bunch of other research exomes or genomes that are just sitting there. We use that as part of our panel of normals.
So you're saying we could use the normals from another project?
Yes, yeah. Correct, yeah, that's what we do. Mm-hmm. Yeah. Go ahead.
Yeah. So we have been debating this a lot. So the sort of campus and structure, the panel of normals that they use includes the whole exarch, and the panel of normals that we use only includes sample sequencing house. Which one is correct?
The one that is that guarantees, or as close as possible to guaranteeing that the samples were processed the same way. Yes. Good.
So let's say you have 100 tumor samples, but matching controls only for, let's say, half of them. So would you want to analyze them all using the panel of normals, or just for the half which doesn't have the matching controls, and for the other use the paired mode?
Use the panel of normals for all of them, because again, it's the matched normal will allow you to eliminate the germline variation from that person, but it doesn't do anything about, like by itself, it doesn't do anything about the technical artifacts that are causing noise, and that would cause a lot of false positives. So that's the job of the panel of normals. So definitely you want to use the panel of normals in all cases. That slide was just saying it's even more important to use the panel of normals when you're in tumor-only mode. So it's like then it pulls double duty, if that makes sense. Any other questions?
Matching normal in this particular case, does it mean that the normal is just not the tumor, but just pick up part of that tissue that is not tumor, or you take it, or is it the case that you take it completely and different individual movement just with
So for the matched normal, it's healthy tissue from the same person. Yeah. Some people will actually tissue match, but we find that just using a blood sample is usually good for all purposes. Any other questions about panel of normals? Yeah, go ahead.
Whilst obviously because the panel of normals
Mm-hmm.
Is there like a rule of thumb for how many is enough?
How many is enough? That's a great question. So our standard recommendation is at least 30, but that's a really low number. Ideally, we typically work with, I think last I checked, was several hundreds, like 500 samples or things like that. As you say, the more the better. But if you have fewer samples than that, like whatever you have is better than nothing. One of the other things that we've sort of experimented with a little bit is, like, can you use, if you have just no normals to use as a panel of normals, can you use unrelated samples? It seems that it's still worth using a somewhat random set of normals, try to match them as much as possible based on what you know of how they were processed. It's still worth using that over nothing at all. Yeah. We do not currently provide a standard panel of normals. There was an idea that was explored of using Nomad to generate a sort of mega panel of normals that would have just everything artifactual seen across everything ever. Ran into some technical difficulties around that because there were just too many sites in it. And the idea was to call it the Common Recurring Artifacts Panel, which spells out crap. So you could literally say you were filtering out the crap, but that effort seems to have stalled. Maybe we'll resurrect it. Let's see. Mm-hmm.
Would it matter to have variable ploidy in the set of normals in the panel of normals?
So you're saying in the case of non-model organisms, what do you mean by including variable ploidy?
So if I have samples from species with different ploidies, or strains of different ploidies, so would I combine them? Would it matter?
So are you looking at doing somatic analysis on those organisms?
Yes.
Or,
So, yeah.
okay.
Yeah. So let's say I have an evolved population, very similar case as a cancer case, but it's just microbial.
Oh, okay, I see.
So then making the panel of normals, could I include samples of strains for which the ploidy could vary from diploid to triploid to?
Are we talking yeast? I trained as a microbiologist. I was like, that sounds familiar. The nice thing about the somatic calling model is that it's agnostic of ploidy. It basically decides, since I don't have a reasonable assumption of what it could be, I'll take whatever comes in. It'd be interesting to actually test this out, but I think that should be fine. Because the model is pretty, like, give me whatever you have. And actually, in some cases, we have sometimes recommended trying the somatic caller for non-somatic calling, but on organisms with, like, if you're doing a pooled experiment. And sometimes we have had questions like that from people sequencing insects, where they're actually crushing together a whole bunch of different individuals and calling variants on the population instead of just an individual. And there you have a sort of pooled ploidy effect. In principle, the somatic calling should work on that. I believe people are using it in that way. Because then you don't have to estimate if you're not sure. I mean, hopefully, if you have a number of insects, you know how many you put in. I'm not sure beyond that. Yeah, anyway, that should work. But try it out, no guarantees. Okay, any other questions? I should move on, sorry. I get enthused about the topic. So that's for the short variants, those are the main principles that are going to come into play, and we'll go over the details on Thursday. There's a lot of interesting details of how you do the filtering and how you evaluate the cross-sample contamination and take that into account. We'll talk about that on Thursday in painful detail. For now, though, I want to give you the overview of the somatic copy number workflow, and I hope not to say anything absurd because Andre is the expert, right? You're the expert? Yeah. So he'll correct me if I say something outrageously wrong. Okay, for copy number variation, this is a completely different, we're going to look at the data in a completely different way. It's interesting. It's a lot of the same challenges apply in terms of the heterogeneity of the tumor and things like that. But at the same time, you're no longer looking for copy number. You're no longer looking at individual positions in the genome and seeing whether there's a match or a mismatch. You're actually looking at how much coverage do you have at any given site. And what does that tell you about whether there might be copy number amplification or reduction? Because if you think about it, when you're doing a sequencing experiment, if you have one copy of a particular region of the DNA, you expect to have, let's say, about 20, 30x coverage. So let's say 30 reads at any given position. That's your coverage. If you have 2 copies of that region within your cell, whether it's 2 copies on the same chromosome or you have a duplication of the chromosome itself, you're going to expect to have twice as much coverage at that site, because most of the time, so those reads are going to align to a single place on your reference, and then you'll just observe twice as much coverage. Or if there's a deletion, like a hemizygous deletion, meaning one copy is deleted and the other chromosome is intact, you'll expect to see half as much coverage. And so what you're going to do is, for a particular target, and here we're talking in the exome case, you have targets. If you're in the whole genome, then you bin your genome territory into targets. And basically for each target, count the amount of coverage you have. Once you have that, and then you look proportionally across the entire genome or exome, how much you have on average, and so proportionally, how much do you have at any given place, right? And most of the time, you should be around 1. So this is, each dot is the proportional coverage value for one target. And it's plotted here. These are all the chromosomes aligned end to end. The color is not meaningful. It's just indicating where each chromosome starts and stops. So we're alternating between blue and orange. And so the y-axis gives you proportionally how much more or less coverage do you have there compared to the average. And what you see is that most of the time it's around 1. And then in places where you have copy number variation, you'll either have a jump or a drop. Okay, that's the basic idea of how we're looking at copy number variation. Now where it gets tricky is that when we're doing the sequencing, there's a lot of factors that affect how much coverage we end up seeing. Because there are some regions where, depending on if you're working with exome, especially depending on the bait sets that you're using, depending on the exome kits, and some other biochemical effects, you can have pretty dramatic variations in the amount of coverage you have for any given site. And so what's really important in this workflow is the normalization step that happens. After we've calculated the proportional coverage, we have this normalization step. And the normalization is going to use a panel of normals, completely different from the short variant panel of normals. Its goal is also to eliminate or compensate for technical artifacts, but it's in a very different way, and the information is very different. And we'll go into that detail on that tomorrow afternoon. But basically, the idea is that you use this reference panel that shows you on average how much you gain or lose in particular regions to normalize the coverage that you have for your case sample. And that allows you to have a much more accurate picture of how much increase or decrease you might have in particular targets. You have that for a bunch of different targets, and then there's a tool that is going to define segments that are contiguous, that have the same amount of increase or decrease in coverage. And then that will call, there's a calling step that will actually say, you have a copy number variation here, here, here. Does that make sense? Okay. And so there's a progression of multiple steps where you go from having raw copy ratios to having proportional coverage, normalization steps, denoising, segmentation, and in addition to that, we can also compute the minor allele frequency of specific alleles. Okay? And we'll go into a lot of detail on that tomorrow afternoon. The panel of normals, I mentioned it already, is made from unrelated samples, kind of the same principle as for the short variant workflow. But the point here is really to do that normalization step, to remove the noise that comes from just technical factors that affect how much coverage you can expect in any particular region of the genome. So again, the other thing that it has in common with the short variance panel of normal is that it's really important to use the same processing. So the same capture kits if you're working with exomes and so on. I would say it's especially important for copy number on exome. Yes. And that is it for somatic variant calling overview. These are the main concepts that are going to come into play. And again, on Thursday, we will do some hands-on and go into more detail into how the algorithms actually work. Are there any questions? No? Yeah?
Relating to the anonymous is your site recommendation for at least 30 and even more possible
Yes, yeah, it is. Yeah, so we also say minimum 30, but again, it's like a, please use more if you can. Yeah. Question?
So what about this form of the number of chances? What about the algorithm so if one is really tiny and small, or if you have two different problems in the cancer?
So you're asking if the
Different subclones are different C and D.
oh yeah. Let's talk about that on Thursday. That's going to get into the detail of how it works. I'll hand that off to Andre too. But I want to make sure we keep enough time for this afternoon for the pipelining. Any more questions? No? If not, that concludes kind of the overview, the set of overviews of the science of genomics and variant calling. Next up, Dan is going to join us. Dan is our guest star, who's an engineer as part of, I mentioned the data sciences platform. He's in one of the engineering groups that develops the workflow management system we use for running pipelines. And so he's going to give you an introduction of how that works and what are the key moving parts that are involved in actually running GATK workflows at scale.